Welcome to this webinar. Before we begin the presentation, I want to provide you with a few housekeeping items. On your screen, you will see a taskbar with icons. Each icon is assigned to a particular element of today's webinar. Click on the person icon to learn more about today's speakers. Throughout the presentation, you can network with others or submit questions to the speakers in the Q&A and chat box next to the slides. Download resources from the cloud icon. After the webinar is over, please take our survey to tell us how we did. Today's event is being recorded and archived and will be available within 24 hours. For on-demand questions or comments, send us an email by clicking Need Help? Email us. If you experience any technical issues today, please refresh your browser by hitting F5 for PC or Command R for Mac. And now I'm excited to turn it over to today's moderator.
Good afternoon. Welcome to our webinar, The New Trend for Recovering Microorganisms in Beef. This event, brought to you by Food Safety Magazine, is sponsored by bioMérieux. I'm your moderator, Barbara Van Renterhem, Editorial Director of Food Safety Magazine. Thanks for joining us. Today's presenter is Terrence Arthur of the USDA Agricultural Research Service. He serves as a research microbiologist in the Meat Safety and Quality Research Unit at the U.S. Meat Animal Research Center. The research objectives for the unit focus on foodborne pathogens and antimicrobial resistance associated with the production of red meat. At the end of our program, our presenter will address as many of the questions that you submit during the presentation. This event is being recorded and will be archived on our website, foodsafetymagazine.com. To get things started today, I'd like to introduce Paula Avery, America's Food Market Manager for bioMérieux.
Hello, everyone. Good afternoon, and thank you, Barbara. I appreciate Food Safety Magazine hosting so companies like ourselves, bioMérieux, have a channel that we can share innovations in food science. And specifically for today's topic, a new trend for recovering microorganisms in beef. And our speaker today, as you mentioned, from the USDA ARS, and for those that are unfamiliar with this governmental branch, they conduct research to develop and explore solutions for agricultural problems. The vision of the ARS is to lead America towards a better future through agricultural research and information. Representing that vision today is Dr. Terry Arthur, who serves as a research microbiologist. And Dr. Arthur has over 80 peer-reviewed publications, and you can check out Dr. Arthur's full bio in the webinar. There's a little link you can look at it. And I'm excited to hear more about this new trend for recovering microorganisms. So on behalf of bioMérieux, I would like to present to you our speaker, Dr. Terry Arthur.
Thank you, Paula, and I appreciate the opportunity presented by bioMérieux and Food Safety Magazine for to come forward and present the research that we've been doing. So today, it's a new system or new methods for beef trim sampling, and as Paula mentioned, I work for the Agricultural Research Service, the USDA, and we're solely research-oriented. We don't have any regulatory responsibilities or anything, so we are just research, and our focus is on red meat. And the other point of interest I want to note here is that I had a colleague that was instrumental. We both really were, we co-developed this technology, and that was Dr. Tommy Wheeler, our research leader here at the Meat Safety and Quality Research Unit at U.S. Mark. And so he's not here today. I'm going to present our research and tell you a little bit about what we've been doing with this and how we see it being implemented. So there are going to be a few polls today, and just to get an idea what our audience, what they're kind of made up. So first one is, what industry are you affiliated with? Beef, pork, poultry, fruit and vegetable, or other? And I'll give you some time to fill that out. So this was developed in beef, but we are seeing applications in other meat industries. And so I think as we go forward, it will start out in beef, but we hope to see it used in the other meat industries, and then potentially in fruit and vegetable as well. So, but today's research results have been, and this is primarily, or this is solely done on beef. So, results have come in, and so we're about heavy in beef and split with other. So that's interesting. That could be in regulatory or some other areas, but primarily beef, and that's what we thought our group would be today, and that's where we've started, is in the beef industry. So a little bit of background. Well, why did we do this? What was the need? So bacterial contamination of non-intact beef. So this is for raw beef trim is where we have focused. So the product contamination with the pathogens, it's one of the biggest challenges in the beef industry. It's what they have to deal with day to day to produce a safe product. And so meat processors, they have a complex system of antimicrobial interventions to make sure that they're producing a safe product. And so in that system, they have interventions and they have good management practices, and they also have a system what they call test and hold. So beef trim is produced, and it's produced into these 2,000-pound combo bins. Each bin can be a lot, so a 2,000-pound bin would be a lot, and they have a test and hold system. So that 2,000-pound combo, if that's their lot, it's tested, and it is not released to commerce until that test comes back as positive. So a robust test and hold system is definitely a part of the food safety systems in the modern commercial beef processing plant. So an effective test and hold process is basically divided into two different areas. Sample collection, so you need representative of the entire lot, so that can be single combo testing is now 2,000-pound lots. For a while it was 5 combo testing, so you'd have 5 2,000-pound lots, but today we're going to focus on single combo testing, 2,000-pound lots. And you want to target the surface tissue on the beef trim. So contamination with the main organism, the main pathogen for beef is E. coli 0157H7, and that's a surface phenomenon. It happens on the surface of the beef carcass, and so when you target sampling, you want to target the surface of that beef trim. And the second part is pathogen testing. So once you've collected sample, you have to test it for the pathogen, and you want that to be specific, so you're identifying only the organism of interest. You're not getting a bunch of false positives, so you're destroying product unnecessarily. And you want it to be sensitive, so it is very robust. It can detect, we'd like to say, 1 organism per sample, so very, very sensitive, so you're not getting any false negatives. You're not releasing product that's contaminated. So today, we're only going to talk about the sample collection side and what is a robust sample collection process and what we've done to kind of improve on that. So there were 2 methods that have been out in the beef industry for a while, and I'll describe those. So one is the first one is the N60 excision sampling for beef trim, and that involves collecting 60 pieces from a lot. So it's either 12 pieces from a 5 combo lot or 60 pieces from a 1 combo lot. And those are targeted to be carcass surface pieces. So the technician that's collecting the sample would focus on collecting samples that have some of that carcass surface on them. They trim a small 1 by 3 inch segment of that, and they'd collect 60 of those pieces. And you can see that's going to take a while. It's kind of laborious, takes a little fair amount of time. And that total sample, the target weight is about 375 grams. A lot of times you overshoot that. So we're talking about a pound of meat per sample is lost on just due to the collecting the sample. So right there you have a fair amount of labor and time, and then you're also losing meat on each sample you take. So the next, that was the IEH out of Seattle came along and developed the N60 Plus sampler for beef trim, and this was to alleviate some of the earlier issues with labor and time. And so it samples down, it kind of shaves down through the material. It doesn't core through that, it shaves surface material, recovers equivalent amount of microorganisms. So when they did the validations, they had a letter of no objection that was approved because they were able to recover the similar amount of microorganisms that the N60 Excision was. And so that was the gold standard at the time. This was equivalent to that, and so they had a letter of no objection, and it facilitated that single combo testing. So it was a little bit, they could do that a little bit easier than collecting the N60 Excision from each, for 60 pieces from a combo. They could do this. So you still had a few detriments, so you still was somewhat time consuming and take labor to drill down through there. The issue is you have dry ice that's put in these combos, and trying to blunt and drill through those dry ice sections got rather difficult, and we were getting some feedback from the beef industry that we really needed to improve that. You also, again, still had lost meat. It wasn't quite as much as the N60 Excision, about half as much, but you're still losing meat on every sample. So there was still room for improvement, and we still didn't think. The goal that we wanted to see is, can we sample all the way through that combo? So you have 2,000 pounds that are filling into that. I've said you either take a pound of that or a half pound of that, and can we get something that's more representative? For a robust sampling system, you actually need to be as representative as possible of that entire lot. And so that was the goal. So another poll question. So what sample collection method do you currently use? N60 Excision, N60 Plus, a rinse, that's more poultry side, a grab sample, typically on the ground beef side, or something else. So go ahead and answer that. So right now, or when we started this particular process, the N60 Excision and N60 Plus were the two main methods. They were really the only two methods that were being used in the beef industry right now. So our method that we're coming up with was something to extend the reach, let's say, and give us a more robust sampling than those two. And that's what we tried to do, something that would sample all the way through the combo, maybe not be destructive, so no meat was collected during the sample, and see if we couldn't just improve on some of these other labor and time issues. So looks like results are in on that. So most people are using an N60 Excision right now. We have a few that are using N60 Plus, and then a few that taper off. So we kind of expected that. The beef industry would probably be, I would have expected those numbers to be about where they were for N60 Excision, N60 Plus, and then, like I said, the rinse is possibly a fruit and vegetable or poultry, and then we have some grab samples that happen primarily in ground beef. But the basis of what we're going to talk about today is to be used in lieu of, to try to give you some labor, time, and cost savings in lieu of the N60 Excision and N60 Plus. And as we go forward in some of the other industries, to possibly be in lieu of the rinse samples and maybe the grab samples as well. So our goal, Dr. Tommy Wheeler and I, were we set out to develop a technology that samples all or as much of the beef trim that falls into that combo as possible. And we wanted something that was non-destructive, so you're not losing meat for every sample you take. We wanted to reduce the labor required. That was a big input that beef plants were having, is a lot of labor to collect those samples. And we wanted something that was rapid. So something that could be, that sample could be collected very, very quickly and would really dramatically reduce the amount of labor that was required to collect these samples. So we started out here at US Mark in our pilot lab, and Tommy and I worked to evaluate a bunch of different methods in our pilot plant. We have a commercial trim conveyor here, and we did a variety of methods to kind of sample beef and see what was going to work best for us. And so we came up with what we identified would be the best method, and it's going to be the continuous sampling device we're going to talk about today. So we had this device we thought would work, or this method we thought would work, and so we needed somehow to further develop that and commercialize this technology so we could get it out to industry. And so we developed a cooperative research and agreement a research and development agreement with AIMTech, which has formed the company Fremonta specifically for this technology. So you see Fremonta's listed there in the lower left-hand corner, and that's one of our cooperators, one of our partners. We filed a joint patent with them on this technology, and it is the continuous sampling device that we're going to talk about next. So once we had the commercial partner on, we went through some further development. So initially we had looked at materials for what was going to be that sampling material. And you'll find out how the whole system works. But there's a sampling material, semi-absorbent material. We needed something at the end of these conveyors to collect the sample. And we started out with the old cellulose sponge. That was what had been used for carcass sampling for years. But it was too bulky for the samples. These belts are typically around 24 inches wide, so we needed something that was going to be smaller, thinner, and the cost was going to be reduced from what cellulose sponge was. So we came up with a spun bond polymer, and then we had to have a sample holder to hold this at the end of the conveyor, so as the meat comes off of the conveyor, it rubs across this, and we needed something to hold that in there. And everything I'm going to tell you about for our data collection from here on was done in commercial plants. So we didn't do it in our pilot lab. Once that proof of concept was done, everything else was done in commercial processing plant environments. And that way we could get feedback from industry as to what was going to work for them. So we really wanted to have something that was a technology they were going to be able to use, not just something that would work, but it had to have such special requirements that nobody could implement it. So we worked very closely with industry, and I think you'll see that as we go along. So this was our initial kind of design development. So you can see the conveyor and the meat coming down the conveyor. And then as it falls off the conveyor, it goes down across what's listed there as a CSD cloth, the continuous sampling device cloth. And at that initial time, it was a 24 by 6 cloth. We've changed that a little bit, and we'll talk about the dimensions as we go along. But what you're getting is each piece that falls across there has a good chance to rub across this semi-absorbent material and collect a surface sample off of the beef trim. And that was our main goal. Could we sample as much of that trim falling into the combo and get a sample that would be reflective of as much of that lot as possible? That's the bottom line, and that's what I think we've kind of developed here. So I'm going to go through a few results. I'm not going to go through all the results at this point in time, but I'll give you a few. And then we have a reference for the published paper that will be in this presentation. You can see that, and that has all the results for these initial trials. So we tried to evaluate this in 3 different ways. So this particular test we did was 100 different combos, so 2,000 pounds of beef trim split between different lean points, so a 50-50 trim and a 93-7 trim line, so we can again make sure this worked for different fat contents in the sample. And we tested for E. coli 015787, the pathogen of interest, and we also tested for indicator organisms, just total plate counts or generic E. coli. And you see in the data on the left-hand side we have a method. We have either the continuous sampling device, and then we have N60 plus or N60 Excision. And we did 100 samples off of each of those. So we had 100 different combos, and each combo got tested by all 3 methods. So the problem with testing for pathogens is usually you get very little data on that because the typical prevalence is about 0.1%. So you see we get a lot of zeros there. And we'll come back to some of the pathogen testing in a bit, but for the average APC, that's aerobic plate counts, there was no statistical difference between any of the methods. Also for the average log of E. coli counts, again, not statistically different. So we were proving equivalence with the standard methods, N60 plus and N60 Excision. Okay, so in the next round, we looked for salmonella. So in this particular experiment, we took 214 different combos and looked at them as either the continuous sampling device or the N60 Excision method. And you can see there that our percent positive, we had 9.2% for the CSD and 6% for the N60 Excision. Those were not statistically significant. They were equivalent and no difference for that level of testing. So again, equivalent to the N60 Excision method. And then the last one I'm going to show here is because we had low levels of pathogens and we wanted to identify could we detect these low levels, and so we needed to kind of investigate how to do that, and one of the ways to do it is to use surrogate organisms. There's some E. coli, generic E. coli strains that FSIS, the Food Safety Inspection Service, recommends for inoculation surrogate testing. And so we used those, and we inoculated in a commercial plant with these surrogate organisms. And for the continuous sampling device in the N60 Plus, we had 20 combos. N60 Excision was 4, and that's because those were 5 combo lots. So we had 20 combos, but they were divided into 5 combo lots, so 4 total samples. But what you see here is that the CSD for both the 50% trim and the 93% trim got 100% of those, was able to recover 100% of those inoculated organisms as a positive. It came back as positive for 100% of the tests. The N60 Plus had 70% and 90%, and the N60 Excision was 75 and 50%. So we did see some significant differences there when we ran the statistics on those. And so, again, we were the same or slightly better than the N60 Plus or N60 Excision. So that's all the data I'm going to go through on the continuous sampling device. But if you have a chance to go through the paper, there's much more data in there. So what is the continuous sampling device, and how does it work? So it starts with a cloth. So our collaborator, Fremont, they produce the microtally cloth. Comes as a sterile cloth in a perforated bag, and so you tear off the top, and you can take the cloth out and apply it. So it comes pre-sterile, and this is what's going to be your sample. So you collect that, the meat collects onto that or rubs across that, and that's your sample. You put it back in the bag, and that bag goes to the lab for enrichment and sampling. So before we move on too far, I'm going to have another poll question about where are we doing our sampling and where is it being processed. So where are your samples currently analyzed? Do you do an in-house lab? Do you have a central internal lab? Or do you use a external reference lab? And so that cloth, as I said, will be put back in the bag, and then it goes to the lab. And it can either go to an in-house lab on site, or it can be shipped overnight. And we'll talk about how we've tested this. We've done both ways. So we wanted to kind of capture what was going on in the industry. Excuse me. So, looks like we have the majority has an external lab, and so that's going to involve some kind of a shipping overnight to get to the lab. And as I stated, when we did our initial testing, a lot of this we did both, where we'd ship it overnight, so it was 24 hours, or it was just sent to the lab right after it was collected and then processed that same day. And so we try to follow along with industry as best we can, and so we've covered both of those situations. So regarding the cloth, so the cloth, we get a little bit of question about what is the cloth, how is it made, and what do you have to do with it. So once you get the cloth and it's collected the sample, you just add 200 milliliters of your enrichment material, your enrichment media. So we tried to make this so it wasn't restricted to any particular platform for detection. So whatever the company was previously using for their meat testing, we had them process the cloth the same way. So we haven't restricted it to any particular media or any particular detection method. So it's not pre-wet. It's put on dry right out of the bag. Put it into the cartridge, and we'll talk about the cartridge here in a second. There's no antimicrobial in the cloth. It's all, the material is tested to make sure there's no antimicrobial in the cloth. It's sterilized when you get it, so it's ready to go. And then the other question is neutralizer. Do you have to add any neutralizer afterwards? And so in all our testing, we did not add any neutralizer to it. No matter where the plant had their antimicrobial, if they were spraying an antimicrobial at the end of the conveyor or wherever they sprayed it, we did not add a neutralizer. And so you'll see, as you saw previously with the data, we didn't have any issues with the neutralizer causing any effects. You could add neutralizer if there's a new antimicrobial that comes along that we determine we may need to add something, but in our studies where we did with most of the plants were using, I think, peroxyacetic acid at the time, we did not have an issue even if we shipped it overnight. So right now we do not recommend or are not adding any neutralizer to it. So the next part of the continuous sampling device is the cartridge. And this is the third, our third cooperator is Sentry Engineering. And so they worked with us to develop the hardware. So we needed a cartridge system. You see we have the green cartridge system, and the green color came from a recommendation from industry. It initially, you'll see some pictures as we go through, that was initially kind of a brown. Our prototype was brown or white, and industry said, no, we want to make sure it stands out in case somebody drops it or something. So we went with the green, and that's been well received by industry. So it's a two-piece cartridge, and you put the cloth on that bottom base plate, and then the top half snaps in place. And you can see there's some recessed areas on that bottom part of the cartridge, and on that top plate on the underside there are some extended areas. So it's a tongue and groove like system to actually put some positive pressure to really force that cloth in there. We designed it to have zero failures. That cloth cannot come off and fall into that combo. So in our development, we went through to make sure there was no failure. There's no way we could have that cloth come off during production and fall into that combo. So that was the goal, and I think we've achieved that. We have run, since our initial test, we've run about another 2,500 tests, and we have had zero failures. The base plate. It's a stainless steel base plate, and this mounts to the end of the conveyor, also produced by Sentry, and this is part of the Pathtect system, both the cartridge and the base plate. And so once the cartridge is put together with the cloth in it, it slides into that base plate, and that base plate is mounted permanently to the end of the conveyor. And so the whole system, it's the continuous sampling device sampler. So we have the plastic food grade cartridge, so it's a molded plastic or injected plastic, food grade, green color, so easily visible. Stainless steel base plate that mounts to the conveyor line, and then the microtally swab that fits in there. So, so that comprises what we call the continuous sampling device. So you can see then that once you have the cartridge, it can slide in from either side. So it's very rapid to remove that and put a new one in, and the idea is you'd have a bunch of preloaded cartridges ready to go. And so as you filled a conveyor or filled a combo, you'd pull out the old one, put a new one in, and continue to run. So the CSD hardware can be sanitized either with a chemical sanitizer or hot water. I think we recommend hot water just so you don't have any residual sanitizer. Rapid change out, so the idea is you'd have preloaded samplers ready to go. And so you'd have multiple cartridges at each fill station depending on what the volume of that fill station is. And so you can just take care of your demand. There's no waiting on any change out. We do 50/50s, which are very rapid combo fills. We can do those with no problem. So the next slide is going to be a video, and there's going to be some, it's just a segment of the video. The full-length video can be seen at the Fremonta website, but this is just a segment to give you an idea how it works. So there's some dialogue that goes with it, and I'll let it play, and I'll talk on the other side.
While the conveyor is either stopped or diverted to another sampling station, slide the assembled sampling cassette into the stainless steel holder from either end. After the cassette is in place, the conveyor line can be restarted or diverted to the new combo for filling. The cloth collects the sample as the combo fills. After the combo is full and the conveyor line is stopped or diverted, use sanitary procedures to remove the cassette from the holder. Individual plants will need to develop specific procedures.
So, you can see it's very easy to slide in and out. That was the earlier prototype with the brown cassette or cartridge. Now it's green, and that base plate was kind of a movable base plate. Now we have them anchored so they don't move. They're very rapid change up. So what we were able to determine based on this was we had developed a technology with the continuous sampling device that, based on the data we collected, our organism recovery was at least as good as the standard methods. And we had much less labor. so it was easier to take the sample. We had much less enrichment volume, so something like an N60 Excision can take over a liter of enrichment media. We only use 200 mils for each cloth, and you don't have any meat loss, so you don't lose any meat per sample. So you're saving about up to a pound of meat per sample. So one of the questions we get is on this is, well, I've got some combos that don't fill with the conveyor. And so we have been working on alternatives for that. And so the first one is the mobile continuous sampling device. And so you have the kind of the theoretical version on the left and a prototype version on the right. And so basically this provided a version that could be rolled up to a combo as somebody is hand filling that combo, and you put it in the hopper, it falls down across the sampling cloth just like it would on the conveyor. When the combo is full, that hopper can be rotated up, the combo pulled out, new one put in, a new CSD is changed out, and again, very little to no downtime on that. So we do have alternatives to that. Also, we've seen some hand-picked combos that people are standing on a conveyor right off to the side, and so we have ideas for that to mount CSDs just off to the side, a little chute, so they can fill those as well. So there's a variety of alternatives. We would like everybody to get to be able to use the continuous sampling device on all their product eventually. The engineering has taken a little bit longer, and so these are coming online piece by piece. The continuous sampling device mounted at the conveyor is ready to go, and the other two are still a little bit in development, but pretty close to being ready to trial. So that still left us with what can we do right now for folks that have some hand-picked combos. And so we went to the manual sampling device. And what this is we took the same cloth from the continuous sampling device and said, can we, if we just sampled basically manually scrubbing the entire surface of that combo, that's going to be a lot more surface area than the N60 or N60 Plus. Will that give us a good enough sample to be equivalent to the current methods? We're not getting that all the way through sampling. And so we needed something that could be an alternative until we get all of the continuous sampling device methods available. And so what we did was we worked on using that manual sampling device of the cloth, a 90-second sampling across the entire top surface of the combo, and it's rubbed across the surface and then inserted. It's kind of a two-part sampling, rubbed across the entire surface, the scrubbing, and then also inserted into the trim to collect that sample. And so just like we did with the continuous sampling device, we did multiple experiments in commercial plants to validate that the manual sampling device was equivalent to the N60 Excision and N60 Plus methods. So the next slide will be a video of the manual sampling with some dialogue associated with that, and again, I'll talk on the other side of that.
Use both hands to vigorously scrub the beef trimmings with the unfolded cloth. During sampling, scrub the top surface of the beef trimmings and push the microtelly cloth into the crevices between the trimmings to sample as much surface area as possible. This can be done two ways. The first is shown here. From the starting location, work around the perimeter of the combo to facilitate sampling the entire combo surface of beef trimmings.
So how did that compare to the N60 and N60 Plus? So initially, again, we tried with pathogens, and so this one we did lean type of 50%, lean type of 80%. We had about 175 combos on both. On this one, we had what would be a day where a higher prevalence than expected for the pathogen, and we just happened to be sampling on that day, so it's a little surprising to see numbers this high. Like I said, typically they're between 0.1 and 0.5, but we were able to get some usable data for this, and we showed that the manual sampling, while there's no significant difference, was equivalent to the N60 Plus on both the 50-50 and the 80-20 trim lines. So on another study using the N60 Excision, looking for E. coli 157 and Salmonella, 182 combos apiece, and we didn't find any 0157 on this particular trial, but with Salmonella we were equivalent around 5%. And then again with inoculated product using surrogate organisms across the 50-50 and the 80-20, we had 25 combos apiece. No significant difference, numerically higher, but equivalent to the N60 plus method. So, as I stated, there's more data in the paper, and so you can look at that as you go. But the data we collected showed that both the continuous sampling device and the manual sampling device were at least as good or equivalent to the N60 and N60 plus methods, N60 Excision, N60 plus methods. So, as far as, the improvements we'd hoped to get, I think we did get them. So the surface area sampled, vast majority of the meat pieces in the combo, I think you can see that from the video as those pieces come down. The majority of them are sampled on the continuous sampling device. The manual sampling device, while it's not sampling all the way through the combo, it's sampling the whole top surface area, about 11,000 square centimeters. N60 Excision is about 1,100 square centimeters, and then it's difficult to calculate, but the N60 Plus is slightly less than the N60 Excision method. So we're getting a lot of surface area sampled, which is really the main goal of a robust sampling method, to sample as much of the lot as possible. So to conclude on the data side, we had 1,400 samples that were included in the paper. And since then, we've collected about another 2,500 that we're getting ready to kind of put that data together. We had industry feedback during the development, which gave us a lot of valuable input. Like I said, the green color so industry could see it, the rapid change out, and just how to make sure that we weren't causing them any delays in their processing. The way we did this was numerous days, multiple companies, multiple processing plants, and multiple lean types to get as much variation as we could to identify were these methods working appropriately. We didn't want to just do them in one plant with one group of people. We did them across multiple companies, multiple plants, and multiple lean types to get as much variation, make sure that what we were seeing, that data was solid. And it collectively, that data demonstrated that the continuous sampling device and the manual sampling device provided the same or better detection of pathogen contamination in beef trim as the N60 Excision and N60 Plus. So where are we now and where do we hope to go? So with that data that we collected from those 1,400 samples, we submitted that to the Food Safety Inspection Service, and we received a letter of no objection for single combo beef trim testing. We also took that data and published that, and so the reference for that publication there in the Journal of Food Protection, my contact information will be later. If somebody needs a copy of that, I can send it to them. And so that paper can also be used with companies as part of their data for their validation on using this particular method. So we continue to work with plants on validation. So even though we've done these large-scale studies, we are very willing to go to individual plants and get them started with their validation so they can show that it works in their hands. We are testing the portable continuous sampling device for these hand-picked combos to make sure, the goal would be to have a version of the continuous sampling device so everybody can use the same method throughout their plant. And then we're also collecting data on variety meats. So there's been some interest on can you use these methods for something other than beef trim, such as offal and variety meats. And so we're in the middle of some studies on that. The manual device, 3 companies are using it for in-house product, and multiple companies are completing validation studies of their own. And we're also working with downstream companies so everybody is on board so that they will accept product that has been sampled with these particular methods. Continuous sampling device, it is under commercial release now. The multiple companies are completing validation studies, and there's interest in other meat industries, and we're starting to set up trials in the poultry industry and the pork industry. But the continuous sampling device, everything is now commercially available. So the contact information, as I stated, Dr. Tommy Wheeler and I are the contact people for validation verification. So if you would like to implement this in your plants or you have any questions about any of this, you can contact us there. If you want a copy of the paper, we can get that to you as well. So any questions you have on the technical side, we would be available to try to answer those questions and help you. The product availability and distribution comes through Fremont for Canada, Mexico, and U.S. Garth Hoffman is the president of Fremont. Looks like, at least on my end, we may have to fix that. We can get the information to people. It's like the slide might not be presenting right. And in Europe and South America, John Pawlowitz with Sentry equipment can be contacted for the sales and supply on that end. So with that, that ends the technical presentation, and I will turn it back over to Paula.
Thank you, Dr. Arthur. Really, really enjoyed your presentation. And if you guys enjoyed this webinar, we are going to have a second series on May 30th, and it's going to be featuring Dr. Keith Belk of Colorado State University. And now I'm going to pass it over to Barbara to field some questions.
Thanks, Paula. And I want to thank Terry first for a great presentation. And now we'll have Terry address some of the questions that have been submitted throughout his presentation. So the first is, do you have any plans to develop a device for ground beef sampling?
So I would say early in development, we're trying to figure out what would be best, because we've talked about people about what can they do on a ground sample. And this device, we don't believe it's optimal, and but we would like to do something more than a grab sample. But right now, I can't say that we have identified how to transition this technology to ground beef just yet.
Okay. How did you inoculate the trim with surrogate organisms on site?
That's an excellent question. So, again, these were approved organisms from Food Safety Inspection Service, and so we went through a letter of no objection to make sure that we were following the guidelines. They didn't want to have any creation of an insanitary condition, so we used very low levels. But the key here was to inoculate small portions of the combo that were going to fill that lot. And so we believe that the E. coli 157 pathogen is not homogeneously distributed throughout there. It's in what we would say non-random, non-homogeneous distribution. And so we wanted to replicate that with our inoculation. So we took a small portion of the of beef trim and inoculated that, and then we took a random number generator to identify which period of time in that combo fill. So if it's a 10-minute combo fill, we filled at 20 different times. So on one time we filled right at the beginning of when that combo filled, so that inoculated product would be down towards the bottom of the combo. And then using whatever the random number generator told us is where we put it in. And so we had product, that contaminant or inoculated product was at the top, middle, and or bottom of the combo. And so that's how we identified when to inoculate. But that gave us what we think was the most representative sampling.
We have another interesting question. Someone is writing that they sample finished product with very low microbial loads and wondered if there's a way for us to use a system like this so that they could use it to sample indicator organisms or pathogens without pulling hourly finished product samples.
So might need a little bit more detail on that one as far as a finished product sample. It almost sounds like it might be a ground sample, but we don't, the method as it's developed now for the beef trim sampling will detect very low levels of organisms. The N60 and N60 Plus were designed to identify as little as one target organism per sample, so per 375, and so we are equivalent to that. So we do have good sensitivity on the detection, but I need more specification as to what the finished product testing would be.
Now, will you be developing this method for other pathogens like Listeria?
So we believe that can be used for any bacterial pathogen. There's nothing special about the cloth method, and we've identified that E. coli 157, Salmonella, and some other non-pathogenic organisms. We can identify those. We haven't done Listeria, but we don't believe that there would be anything special about these particular organisms. We believe that it should be able to sample any bacterial organism that you may want to.
Great. Have you heard of any interest by the Canadian industry in utilizing this method?
Yes. So we've had some initial discussions with CFIA, the Canadian Food Inspection Agency, and we've shared the paper with them, and so we're working with them to find out. It sounds like their approval might be a little bit different than the Food Safety Inspection Service, but it shouldn't be a problem moving forward with them. But yeah, we are in talks with them to make sure that they are on board with, and I don't want to necessarily say their approval, but something similar to FSIS with their no objection.
Great. We have another question. What is the inoculation level of surrogate E. coli?
So that's a good question. I'm trying to remember off the top of my head. As I said, we wanted to keep it very low and keep it so we didn't have any insanitary condition. And so how we did it was to basically so we have a 2,000-pound combo, and we were shooting for, I think the easiest way for me to do this is to actually just pull it up here. So since we had 900 kilogram combo, we used 1/100th of that was what we inoculated. And so we shot to inoculate at 2 log CFU per gram of that. And so in the entire combo, we were shooting for a 1 log CFU per gram overall. So it was, but we don't think about it as being homogeneously distributed. We believe it was probably, a much smaller area. So it was probably a little bit higher concentration in a localized area, and that's kind of how we think of the E. coli 0157 would be as well.
Great. Now, do you see this type of methodology being developed for fruits and vegetables, for example?
Yes, and our partner Fremanta is working on that. Our research objectives here at the U.S. Meat Animal Research Center don't include fruits and vegetables. We're happy to transfer that technology, and so Fremanta, our partner, is kind of they're the ones that are doing that.
Great. Now, could the microtally cloth be used to sample larger carcass surface areas than traditional sponges?
So we are just going down that path. We've just started looking at carcass sampling and what are the possibilities, and specifically for that reason, because the typical 3x1 cellulose sponge and doing the 100 square centimeters, we're looking for something that could do better. And we believe that this may be able to do that, but we don't have the data yet to say for sure that it will.
Okay. Here's another question. Are you using an analytical method equivalent to the FSIS MLC5C?
So the way we've done this is my answer is essentially yes, but it has to be qualified. So the way that we did it with most of this data is we worked with the industry partners and the beef processing plants, and so the methods that they were using. And so they are, as the MLG lists, that you have to use a particular method or something equivalent. And so while we used a variety of methods, they would have all been equivalent to the MLG, but we didn't follow the MLG's specific method throughout this process.
Okay, that makes sense. Now, could someone invite Fermenta or Sentry to their plant to see what the best application might be?
Yes, yeah, and that's perfectly fine. For those purposes, I think, and go ahead and contact Fermenta or Sentry or us at the U.S. Meat Animal Research Center, and we'll figure out who's the best to go out and do that. Between the three different groups, some of us have different specialties, and so, yeah, contact any one of us, and we'll figure out who needs to go out and figure out what's going to be best in your plant.
Great. Now, I had posed a question that someone asked about fruits and vegetables, but someone else asked about applications for low moisture products like cereal grain.
That one might be more difficult. I mean, I think we'd be willing to try and see. I mean, the best way to do some of these is just do side-by-sides with the current methods and see if they are equivalent or not. Low moisture, just unless it's in the kind of the dust associated with the product, might be a little tricky. So I'd hate to say that it would or wouldn't. I'm a little bit doubtful, but it'd be best just to do a side-by-side.
And another question is, what if you're filling totes or boxes? Is there anything else that needs to be done?
If you're filling boxes, so that's where we believe the manual is, and that's where our work is right now on products like the offal product or even boxed beef trim, is that we can take the manual sample and it will be similar to the current method of sampling, and either that's a grab method or some kind of a variation on the N60. And we're in the middle of some trials on that right now. And so once we have all that data, we will try to get that out to industry in the form of a publication, so again, it can be used. But we believe it should be fine for that, but we need to collect the data.
Great. Now, one question that came to mind as you were speaking for me is, you're passing a lot of the trim over this piece of cloth. Have you looked at how soon that cloth becomes saturated, so that maybe you're kind of skewing the sample to what first runs over the cloth as opposed to the end pieces of trim?
So I'm going to answer that two different ways. So first of all, the way we look at it is not necessarily as a saturation, but it's going to achieve a steady state. So remember, we're looking for plus minus. This isn't something that's going to continually collect, the bulk of the cells, and so you're going to get that. But it's more of, these are prevalence tests or qualitative tests. So you're looking for a plus minus. And the way I look at it is, as product comes over, say late in the fill, you still have the ability to remove something that was on the cloth and pick up new. And so that's why I think when we did our testing, we looked at it, one, is just compared it to what was the traditional test, so the N60 and N60 Plus, and our feeling was if it is able to collect that, we should be able to get, be equivalent to that particular method. The second way we looked at that is in that inoculation. So in a lot of those samples, we inoculated at the end of the fill. And as you saw, we got 100% of the inoculated samples or the inoculated combos came up positive. So we were even able to pick up those at the end of those combo fills. So we've tried to look at it a variety of different ways, and that's the way I would say it probably comes in. that we're doing fine at the end, but it should be thought of more of a steady state operation and not a saturation.
Sure. Now, you said that at some point you did a lot of the experiments directly in commercial plants. So did you have any surprises once you moved to an actual plant situation as opposed to your pilot studies?
I wouldn't say we had a lot of surprises. I mean, our group, the Meat Safety and Quality Research Unit here at the U.S. Meat Animal Research Center, works quite extensively with industry. So we're up in the plants a lot collecting samples of different varieties. And so we are pretty familiar. And we didn't, as we developed this, we have that dialogue with industry, kind of telling them, the initial ideas, how we were planning to implement it, and we took some feedback from them. But I don't think we had any real surprises. The biggest thing is just how different every plant is. And so how can we accommodate this kind of a test to all the different forms in which beef gets into a combo bin? And so that's been the biggest challenge, I'd say. It wasn't a surprise, but it's a 24-inch wide sampler, and so it's ideal for a 24-inch wide belt or even something bigger that you can funnel down to 24, but we've used it on 12-inch lines, and the data is equivalent to N60 and 60 plus off 12-inch lines. So I think there hasn't been as many surprises as just challenges in how to best evaluate the methods. We want to make sure we cover all the different variations so we aren't surprised.
Great. So I think that's all the time we have for questions right now. Please join me in thanking Terry Arthur for his presentation, as well as our sponsor, bioMérieux. If you have any additional questions or comments, don't hesitate to click the Email Us button on the console. And as you exit the webinar, a survey will pop up on your screen. We'd appreciate your detailed comments. Please visit our website, foodsafetymagazine.com, for an archive of this presentation, as well as information about our upcoming events. We appreciate your time today and hope you have found this webinar to be a valuable experience.